Tu fais du sérieux, tu veux pas. C'est là-bas.
Ah non.
Je remplace avec le
Bravo. Et c'est la dernière belle fois. Et une bonne présentation. Tu vas faire du bien.
Oui, il m'a demandé de dire c'est bon. Oui, il a été juste avec le côté de l'aide de Jean le ah, non, bah, oui, le dos, c'est pas bon. Pas trop sens. Donc on va débuter, on va commencer. D'abord, merci Rachid d'avoir invité pour participer au jury de thèse de Mohamed BERRIMI. Avant de te donner la parole, donc je rappelle à peu près 45 minutes pour la présentation. Ensuite, on passera aux questions et pour approfondir un petit peu les connaissances et voilà, répondre à certaines questions de tes travaux. Donc, avant de te donner la parole, je vais présenter un petit peu les membres du jury. Et je vais commencer par les rapporteurs. Donc, par ordre, comme c'est écrit, ADEL Mouloud, professeur, Université d'Aix-Marseille. Alice OTHMANI, MCF-HDR, Université Paris-Est, Créteil. Ensuite, pour les locaux, on a Diarra Pan, qui est maître de conférence à l'Université d'Orléans. On a Sylvie Treuillet, qui est professeur des universités, l'Université d'Orléans. Et on a le directeur de thèse, professeur Rachid Jennane, de l'Université d'Orléans. Et moi-même, CHETOUANI, professeur de l'Université Sorbonne, Paris-Nord. Donc, voilà. Maintenant, vous avez 45 minutes pour votre présentation. En anglais, je suppose,
ouais.
c'est ça?
Oui, c'est en anglais, ouais.
Donc, je précise, les questions, bon, je pense que Mohamed parle bien français aussi. Donc, on est tous libres de poser des questions. J'ai oublié, pardon, professeur Mohamed JARRAYA. Je ne sais pas comment le titre, on dit, c'est quoi? Hein?
Medical doctor, I think.
Assistant professor.
Associate professor de Harvard Medical School. Donc, il est en ligne avec nous. Donc, il interviendra également dans la séance de questions par la suite.
Thank you.
The floor is yours.
Thank you so much. Thanks, Mr. President of the jury. Hi, everyone, and welcome to my PhD thesis defense. So my PhD thesis is entitled Novel 3D Deep Learning Models for Knee Osteoarthritis Prediction and Diagnosis using MRI Data. The thesis is part of ANR Mimosa project, and it's supervised by Professor Rachid. Before I start my slides and presentation, I'd like to take a moment to thank all the jury members for accepting to review my PhD work, and for all their valuable feedback and comments on my manuscripts, and also for coming all along here for the discussion. So my presentation today is in three main sections. First, I'll briefly mention the context behind knee osteoarthritis, what's knee MRI, and what are the challenges that faces AI and deep learning researchers into processing and analyzing knee MRI data. Then I'll list the contribution that we made in the past 3 years in order to contribute to improve the performance of deep learning models for knee osteoarthritis detection, and finally a conclusion. So knee osteoarthritis is a degenerative joint disease that is characterized by the gradual breakdown of the cartilage, and it's one of the main causes of disability around the world. It is unfortunately very painful, and there is no cure or treatment for it, especially when it has progressed. Additionally to this, the knee joint is one of the most complex structures in the human body. In medical practice, it requires detailed and precise analysis to assess different components of the knee structure. Additionally to this, it is a global and a worldwide problem because it's affecting millions of people around the world, causing them severe pain, reducing mobility, and unfortunately, for many cases, full disability. Additionally to this, the economic burden and impact of knee osteoarthritis is severe and is significant. It's costing billions of euros annually for many countries only to develop the infrastructure for the treatment of a diagnosis. In clinical practice, we have two main screening protocols for the diagnosis and for the analysis and grading of the knee osteoarthritis. Mainly, we use X-ray. X-ray are two-dimensional and mainly show bone images, but they are quite limited into showing detailed information about the knee joint and about other soft tissues. However, we also have MRI, which are three-dimensional, but they produce detailed images about the knee joint and other soft tissues as well, such as the tendons, ligaments, meniscus, cartilage, and so many things. So the 3D nature of MRI data makes it really valuable for assessing the knee joint. In MRI, we usually have from 3 angles or 3 views of the scan. We have the axial, the coronal, and sagittal views, and each view and MRI parameters has different sequences or acquisitions, and depend on the acquisition,
like we have different number of slices, like each scan is volumetric and each one has number of slices. And the more we go into the depth of the slices, the more detailed information we can see about different components of hidden soft tissues. For example, as we can see in the animation. So this 3D nature of knee MRI makes it really valuable for analyzing the knee, and also makes the gold standard. However, when it comes to when deep learning AI in general, it's not really easy to apply them direct. So before I start my PhD, I was looking for research papers on the field of knee MRI analysis. And the first thing that I have noticed is that we have limited research papers in this area compared to other medical imaging analysis tasks. And this is mainly due to the complexity of training 3D deep learning models. Also, the limited data resources that we have for knee MRI compared to other injuries. So among all these research papers that we found that they are single view limited, meaning that they train the deep learning models only using one single view MRI data, meaning that they use only sagittal to train their neural network, or coronal, but very often rarely that they combine all the views into a single network. And this is quite limited because for many cases, to have a holistic view of the image of the knee, we need to analyze the knee from different angles and from different views. Additionally, most of the research papers use only single modality. They use only X-ray, mostly X-ray, or very often they use MRI, and then they don't combine both modalities. And last but not least, the processing and the analysis of the MRI is really complicated. Also, the extraction of region of interest is quite challenging. So the overall objectives of my work is to train efficient multi-view deep learning models that are three-dimensional to help improve the performance of deep learning in knee MRI analysis and for also knee osteoarthritis detection. We also want to train efficient models for other knee injuries analysis. We don't just focus on knee osteoarthritis, but we also interest in other injuries. We want to study the effectiveness, which models are better for MRI analysis. Are they three-dimensional or only two-dimensional are enough? And also we aim to propose efficient segmentation and region of interest extraction techniques. So to achieve all these objectives, we have presented four main contributions in my PhD. So along all the presentation today, I will go one by one. So the first contribution is entitled Multi-View Semi-Supervised Models for Knee Osteoarthritis Detection. So this is from our research paper. And first, I will talk about the database that we use, which is one important aspect in my work. And then I will list the main contributions into this chapter. So in my PhD thesis, I use the Osteoarthritis Initiative dataset, which is a multi-center database and has clinical information data about 4,700 patients. And each patient in this database has X-ray of one of his knees or both knees, and also MRI scans, and also clinical data such as age, BMI, etc. So our main focus in this PhD is to use the MRI data from the OAI database. So in order to use the MRI data, we needed reliable ground truth labels to label the scans, and also to train our models for the classification and for the detection of knee osteoarthritis. And for that, we used a common ground truth label, which is called MRI osteoarthritis knee score, which is a scoring system developed by expert radiologists. And it's mentioned that an MRI scan that's considered as osteoarthritis is when it has osteophytes and cartilage loss. So we went all the database, and we could only identify 450 scans that meet the definition. We take also extra 450 scans that are normal cases. So meaning that only 10% of the database is actually labeled. So the overall motivation of this work was actually, can we leverage a deep learning model to be trained like on both like labeled and unlabeled scans. And for that, we propose the following methodology. So we propose a pre-processing step, and then a training strategy and model architecture. So I will list them later. So first, let's talk about the pre-processing step. So the first question is why we want like to process the MRI scans and not just like feed them directly. So the first thing is that we did some data analysis over the MRI scans, and we found that each MRI scan is volumetric, meaning that each scan has multiple slices. However, for each MRI scan, for the slices, like 10 to 20% are irrelevant. And what do I mean by irrelevant is that they don't show any information about the knee joint, as we can see here. So here, for example, we see information about the knee joint. We can do some assessment, but here they will only add some noisy features to the neural network. So we set a range on which usually these relevant slices occurs for each scan. So after setting this range of relevant slices, we went looking for the region of interest detection. So knee osteoarthritis actually only affects the knee joint area. So all the other areas in the knee MRI are actually relevant, and they will only like include more noisy features to the neural network training. So we set also the area in which usually this the knee joint usually occurs in the MRI slices. After setting both a relevant slices range and our region of interest areas, so we processed all the MRI scans in our database according to these settings. So just to recall, so just we have like pre-processed so relevant slices here. We remove them, and also we crop the relevant slices. So now we move forward to the model architecture. So for that, we propose 3D CNN gap. So this is actually a new architecture that is based on residual connections, and in which we train in a three-dimensional way. And why? Because the data here is three-dimensional. So we kind of took a ResNet model, and we converted all its layers to be three-dimensional. So it supports the input data. And then we do some customization. So for each sequential blocks, we train a 3D convolution block that has convolution layers, pooling layers, all are 3D. Then from each 3D convolution block, we extract an identity block using a residual connection. And then we combine these features along the next predistant convolution block. Additionally to this, we conserve the local features from each 3D convolution block into a global average pooling. And then we combine them all at the end. So, and then we pass all these features into multi-layer perceptron for binary classification. So the idea here was to propose a novel model, and then we will compare it to some baselines at the end. So here the idea was, can we train 3D CNN gap model using labeled and unlabeled MRI data? And that's what we propose using the following semi-supervised network. So this is done in 4 steps, but we do it in multiple loops. So the first thing is that we train what we call the teacher model. We take our 3D CNN gap model, and we train it on the labeled MRI data. Then we make sure that this model is quite optimized and really performant. And then we take this model now that's now pre-trained, and we make some predictions on 250 scans of unlabeled MRI scans. Then we generate the pseudo labels of these MRI scans based on the output probability of each scan, of each predicted scan. And fourth step, final step, we merge both pseudo label and labeled MRI scans into one data set, and then we train our model again, and we make sure that's optimized. And then we do this loop up until we label like 4 to 5 times the number of initial scans. And we do that for multiple MRI acquisitions. We do it for sagittal desk, for sagittal intermediate weighted, and for coronal reformat. So let's just go back to our 3D CNN gap. So currently, this model actually takes as input only sagittal MRI scans, but this is quite limited as we need a holistic view of the image of the knee. And for that, we train our 3D CNN gap model using multi-view setting, meaning that we are going to train it into multiple branches, in which each CNN branch focuses on learning unique spatial and structural patterns from its specific view or sequence. And then finally, we concatenate and concatenate and all the features extracted from multiple CNN branches, and we put them for classification. So now let's go forward to the experiment. So we take our model and we train it only on sagittal disc acquisition, then using TSC acquisition, then coronal reformat acquisition. Then also we do some combination between the sequences and between the views. Then also we compare each model within each corresponding sequence using the data before we do semi-supervised and the data that we do after semi-supervised learning. So this is the original data and this is the expanded data. So the results show that using multi-view, multi-sequences, and using the data after we do semi-supervised learning actually gives the better performance than all previous experiments up to an AUC value of 93.2%. We do also comparison within state-of-the-art works, and also we have achieved the best classification AUC, also with our model on the large database, multi-view MRI database of knee MRI. Additionally to this, we made also another contribution in which we take 3D DeskNet and InceptionNet. So basically these are ImageNet based models, but they are 2D. So we converted all their network into 3-dimensional, and we trained them like on multiple sequences and multiple views, and we compare them using our custom CNN model. So our CNN model achieves the best classification accuracy compared to all of them. Additionally, we want to do some experimental results to show if our model is focusing on relevant and important regions of the knee MRI slices or scans, because they are 3-dimensional. So we have high attention in joint regions, meaning that the model is really paying more attention to the knee joints in which like the changes actually occurs. And also we have like from different planes, from different acquisitions, we have consistent attention, meaning that our model is not view or sequence biased. So as a conclusion, so here in this contribution, we proposed a 3D CNN gap model and we train it into multi-view setting. We also proposed a pseudo-labeling or semi-supervised network to expand the number of scans of our database. We have seen that the multi-view aggregation forces the model to rely on patterns that are commonly across different views or different scans, rather than offer emphasizing any single view features. We have seen that expanding the data using semi-supervised learning model boosts the performance of the CNN model that we propose, and also multi-view based models are better than single view based models. So now we move forward to our second contribution, which is entitled M3Net. So M3Net is a multi-view, multi-model, multi-label network for knee injuries analysis. This is from our research paper, which is now under review. And actually, the motivation of this work is as follows. So we have seen that in the literature review, that most research papers use single label models, meaning that for different injuries, researchers tend to train different models for each specific injury. However, we can just train one network for different injuries. Also, they use single modality, as I explained earlier, and also they train their models on very limited data, which poses so many questions about the robustness of their networks or their models. So for this, we propose a novel neural network model that can do classification and detection of two injuries that are more likely to develop knee osteoarthritis. The injuries are meniscus lesion and cartilage lesion. So the idea here is to train a neural network to classify both of them and detect both of them. And so we want to train this network on a very large database. So just to recall the pre-processing step again. So in the first contribution, we removed the irrelevant slices and we did some cropping. However, this is quite limited because we know that not all MRI scans in the database has the same parameters, maybe. So basically number of slices, knee orientation can vary from one scan to another. So like setting a predefined range or predefined region of interest is quite very limited. And also the region of interest could vary from one scan to another. So, and it's fully semi-automatic, like we need to do some manual settings, then we do another automatic setting. So this is quite limited. So for this contribution, we aim to enhance this pre-processing step, and we propose the following custom level V8 model. in which it has three main steps. So the first step is that we do, just to recall YOLO. So YOLO is state-of-the-art object detection model, powerful enough to detect more than 1,000 objects in the nature. However, it's quite limited when it comes to medical data, and more specifically to knee MRIs. So what we do here is want to do a fine-tuning of YOLO V8 in order to detect the knee joint in the MRI slices. So for that, we do some manual labeling here, like we take some data and we manually label it for YOLO. Then we fine-tune YOLO for these images. So the weights now of YOLO V8 are adapted to recognize the knee joint areas. And then we do some validation of YOLO. We make sure that's really optimized. We add some data or remove it. And then, so here basically, YOLO V8 is purely two-dimensional. However, our data is purely three-dimensional. So for that, what we do is that we take from each MRI scan, we extract all the slices, we put them one by one, and at the end, we aggregate all the slices to form one cropped and pre-processed MRI scan. So we take these MRI scans and then we train the following network. So let's depict this into small details. So at the beginning, we have like the MRI and X-ray scans of one single patient, of one single knee. We put all of these data into separate branches, CNN models. However, for the MRIs, we train a 3D ResNet, and for X-ray, we take 2D ResNet. And we extract the features from each modality and from each view, MRI view, that corresponds to the meniscus and to the cartilage. And then we concatenate the features of both meniscus and also the cartilage into separate embedding features. Then we put these embedding features independently to a multilayer perceptron. However, this multilayer perceptron will do binary classification for meniscus and also binary classification for cartilage. So the output here will be 0-1 for meniscus. Is it like torn or good meniscus? And also the same for cartilage. And then we back propagate the weights of M3net until the level of the convolution layers. And then we update all the weights of the network. So this network is actually multi-view because it takes as input coronal, sagittal, and axial. It's also multimodal because it merges and combines both X-ray and MRI features. It's also multi-label because it does two binary classification, and which we call M3net. Additionally to this network, we study different tools or different ways in how to merge or how to fuse our features. And for that, we study linear fusion, concatenation, averaging, also multimodal attention. And then we calculate the accuracy and AC values of both cartilage detection and meniscus lesion detection. And we achieve accuracy up to 97% for cartilage and 87.39 for meniscus. Also, we did some ablative study on M3net to see is really combining MRI X-ray good. And we've seen that MRI is better than X-ray, and combining both of them, like multimodality, is better. Also, we have tested like the network using only sagittal, coronal, or axial, like separately, and then we combine all of them. Like the combination of multi-view is much better than any single view based models. And also we do some task ablation. So as a conclusion, so here in this contribution, we propose a M3net model, which is multimodal, multi-view model that depicts the clinical use and tends to give better results. So M3net is trained on a very large database. And also we employed YOLO V8 for efficient region of interest extraction, improving the feature extraction accuracy. And M3net demonstrated strong results for extracting features and the classification of both injuries. Now we move forward to the third contribution, which is MVMI, which is from our two research papers. And in the motivation of MVMI is as follows. So far we have trained so many 3D deep learning models. However, the complexity of training a 3D deep learning model is really painful. It's really hard, really complex, requires a lot of time, a lot of energy, and lots of adaptation. And however, when we compare 3D deep learning models with 2D deep learning models, 2D deep learning models are much easier. actually, if we compare a 3D deep learning models trained on the same number of scans, 3D deep learning models have 3 times weights. It requires 4 times the number of hardware requirements, so are the GPU. Also, they are 5 times much slower. And the idea maybe here is that let's just train 2D CNNs over MRI slices, but we know that the 3D nature of MRI is really informative. So we kind of want to keep the 3D MRIs, but we also want to train 2D CNNs models. And actually, that's the idea here. And the question was, can we train 2D deep learning models over 3D MRI data? And that's what we try to focus and propose on the following methodology. So in this methodology, we have 2 main contributions. So the first is the feature extraction and also the multiple instance learning. So the idea here, we want to propose a CNN model that's powerful enough to process the MRI scans, and then I will detail what we propose on multiple instance learning. So for the feature extraction step, we propose RescueNet, which is a ResNet model into we introduce some customization. This customization is a squeeze and excitation block. And what does it do? It's actually is that it takes the spatial features, it summarizes them into a global average point layer, and then it puts this vector into a small neural network. And why is that? We kind of want to learn which slices in the MRI scan that has more informative features. And the slices that has more informative features, this simple neural network, it will give them more weights, and less informative features, as scan slices, we are give them less weights. And then we train this network into multiple instance learning. So multiple instance learning is designed to classify instances where class labels are associated with set of instances. So this is widely used for many medical imaging databases, but not really used into an MRI. So we want to do here is that we are going to, as input a 3D MRI scan, we are going to input it into a neural network. So in the neural network training, not as a pre-processing step, we are going to extract the slices of this MRI. And for each MRI is going to act as an instance, and all of them are bag of instances or batch. And then we take this batch and we put it into our RescueNet model. And then we are going to output the output embedding feature of each MRI slice. And then this is the instance embedding. And when we do also and we aggregate the features, the embedding features of all the MRI slices. So here at the input, we have 3D MRI data. At the output here, we have a single feature vector that corresponds to this 3D MRI scan. But in the middle, we train 2D CNN over 2D MRI slices. And this is actually helped us a lot. So this also allows us to make, so since we can learn the instance embeddings of each MRI slice, it can allow us to see which MRI slices are more informative. So at the beginning, we trained as it is, but using only one single view. But we want to go further. We want to make it better. And we propose MVMI. So MVMI is multi-view, multi-instance framework. We take as input sagittal scans and coronal scans, train them into separate branches of multi-instance, and then we aggregate or concatenate the features, both sagittal and coronal features, and we put them into multiplayer perceptron. So for the experiments, we take our RescueNet, we compare it with DenseNet and InceptionNet. We do experiment using coronal only and sagittal only. Also, we combine both of them, like both scans. Also, we train these networks using multi-instance learning, but also without multi-instance learning. So in multi-instance learning, in the instance-based models, they are 2D dimensional because multi-instance learning allows us. And in volume-based, we train them only all 3D dimensional. So the idea here was to see the performance of 2D CNNs over 3D CNNs. So the experimental results show that our model gives better performance when having like multi-view MRI data and also using the multi-instance learning with an accuracy and AC value up to 94.52%. Also, we compare our results with the state of the art, with also our previous work, and we have achieved the best and the highest AUC classification value. So as a conclusion in this contribution, we proposed a new approach using multiple learning to better detect knee osteoarthritis from MRI scan, achieving very high accuracy. In multi-instance learning, the model learns which slices contribute to the overall label by finding patterns across the slices. Our RescueNet model, when trained using multiple instance learning, is much better than when it's trained on the volume-based. So 2D RescueNet is better than 3D RescueNet. And also, multi-instance learning allowed us to train simpler 2D architecture by passing the high computational load of full 3D plenary models. So far in the previous three contributions, we have seen that adding more labeled data using semi-supervised models achieve and boost the classification performance. We also have seen that extracting the region of interest gives much better classification performance. We have seen that training 2D CNNs much better than training 3D volumetric models. So the idea here was, if we So the idea here was, if we train more data, like we combine all these conclusions, if we have more training data and better region of interest extraction technique, and also train better 2D CNN models, are we going to have better performance? And this is what we all want to answer in the fourth contribution, which is entitled Multitask Learning Methods for Knee Osteoarthritis Detection. So here the idea is, we want to propose a multitask learning method, and for that we require to do some data curation, because we need some data to train multitask. And I will explain what's multitask. This is from our research paper. So for that, we do some data curation. So in this contribution, we are solely interested in the 3D sagittal 3D DES MRI sequences, in which each MRI volume has 160 slices. We found a Zeus Institute Berlin lab database in which they have manual segmentations of masks of the 3D sagittal DES acquisition from the OIA database. What we do here in our study is that we want to extract the MRI slices from the MRI scans, and also the MRI slices from the masks. And also we do mask alignment, meaning that each MRI slice is going to have its corresponding MRI mask and also its corresponding label. But then we do some data cleaning, and the overall database that we constructed is total images of more than 211,000 images, in which 105,000 are MRI slices, and also with the same MRI slices have the corresponding MRI masks. So we take this database that's quite balanced, and we do three main experiments. So in the first experiment, we want to train a 3D single task classification, meaning as we were doing at the beginning, like we take 3D MRI scans, we put them into 3D CNN, and we do classification. We want also to do 2D CNNs, like now we have the 2D MRI slices. We take the slices, we take the same CNN, but it's now two-dimensional, and we do classification. But we also want to do multitask, and this is what I mean by multitask. So in multitask, we have MRI scans, MRI slices, their corresponding masks. We put them into a single neural network, which is an encoder-decoder. But here we are going to have two tasks: segmentation task and classification task. So in this network, we have the classification pathway, which is an encoder VGG16 model. So we are going to extract from the encoder a global average point, and we put it into multidiscrete perceptron for the classification. We also have the segmentation pathway, which is as follows. So this is the decoder part, but we also made some residual connection between the encoder and decoder. And also we share the weights between the segmentation mask task and the classification task, meaning that the model will learn how to segment, and also we extract very informative features that we are going to use for classification. So also, as I mentioned earlier, we have segmentation task and classification task. So this is multitask. So now we move forward to the experiment. So in the experimental part, we do some model comparison between of the number of weights and the model size of 2D single task and 3D single task and also multitask models. We have seen that our 2D CNN models are much lightweight compared to 3D CNN models when trained on the same scans, but one is 3D dimensional and another is 2D. I've seen that our multitask network, our model, have achieved the highest accuracy for the classification of osteoarthritis of an accuracy of 96.7%. So also we compare our result again with state-of-the-art, with this state-of-the-art are 3-dimensional, also 2-dimensional, and single task and multitask. And our models are achieved like the highest classification accuracy for knee osteoarthritis detection. So as a conclusion in this part, so we proposed a multitask model for knee osteoarthritis detection using MRI slices. We compare multitask and single task models, like we trained on the same scans. One is type 2-dimensional and the other one is 3-dimensional. So the multitask learning approach segments relevant knee structures while detecting knee osteoarthritis using shared representation across these tasks for improved performance. So these experiments have been conducted on a very large database. So this largest database that exists for knee MRI is composed of 211,000 for both MRI slices and their corresponding MRI masks. So this study achieved the highest osteoarthritis detection accuracy from MRI slices, surpassing state-of-the-art, including those based on 3-dimensional data. So by focusing on 2D MRI slices, this method presents a simpler, cost-effective alternative to 3D models, minimizing the complexity and reducing the impact of noisy features in training. So as a conclusion of all the contributions, so we have used and developed multi-view MRI networks that are 3-dimensional. And these multi-view MRI networks are actually offering full assessment of knee injuries compared to single-view analysis. Multi-model approach in M3net combining MRI and X-ray for multiple injuries classification actually give much better performance than relying only on MRI or X-ray. Training 2D CNNs or 3D MRIs using multiple learning strength the feature extraction and leading to better classification accuracy, and precise and different extraction technique using Euro V8 is much better than the first the feature of extraction. So multitask learning on 2D MRI slices allows models to learn various injury aspects simultaneously, improving the diagnosis precision. So in future work and perspectives, we want to focus on testing our models on broad databases for knee MRI and also for various diseases to ensure broadly applicability and robustness of all our models. We want to explore advanced techniques to reduce the feature redundancy in multi-view and multi-model training. And also we aim to train multitask models for MRI and also MRI reconstruction models. We want to investigate recent models such as vision language models and segmentation models such as segment anything model to expand the capabilities in medical image analysis of MRIs. So for publications, in my PhD thesis, so in the past 3 years, we have managed to publish 4 journal papers. 1 is published and 3 are under review, currently under review, and we have 3 conference papers. So this presentation sources, and thank you so much for your attention and for your time.
Merci Mohamed pour ta présentation. Donc on va passer maintenant à la séance de questions. Et traditionnellement, on commence par les rapporteurs. Donc je vais commencer par Alice OTHMANI, donc je répète, qui est de l'Université Paris-Est Créteil.
Thank you, Mr. Head of the jury, for this presentation. Thank you so much, Mohamed, for the very clear presentation and for the clear thesis. I enjoyed so much reading the thesis. I think I have already explained in my report.
Yeah.
So it was also the same for the presentation today. It was very clear. I want also to thank the head of your thesis, Mr. Rachid, for this very excellent research work. I hope that the comment publication will be accepted. Hopefully, yes, it will be an amazing achievement by the end of this PhD. So I will have a few questions. The first one is related to the generalizability of the proposed approach. Actually, you have evaluated your work in one data set, which is the UAE database, exactly. So my first question, why you have not tested the generalizability of your approach in other data sets?
Yeah. So actually, this is listed in our future work, and this is very important step because the generalizability of the models is very important in deep learning research. However, this is quite limited, as I mentioned in the first challenges, that we don't have too much MRI data, and more importantly, we don't have the ground truth labels. So the ground truth labels that we are currently using are reliable ground truth labels that have been presented by professional experts from University of California in the United States, and which is MOX, and the MOX labels that exist only for the OIA database and only for 450 scans. So this is quite limited to use like other scans. So this is for knee osteoarthritis. However, we also focus on knee injuries such as the meniscus and the cartilage. And we found one database, which is MRNet from Stanford Lab. And this database has only like for meniscus labels. And they don't have like for cartilage, for ASL, for different injuries. So we couldn't kind of like compare our network with their corresponding labels on their corresponding scans. Yeah.
So it was
Yeah, so there are kind of some public database, but the labels we don't have. Yeah, we only have like 450 scans that are currently labeled.
But you have
Yes.
is it possible to use this data set to test the first proposed approach?
Yeah, actually, that database is for the segmentation masks only. So this, so they take the OI database and they did manual segmentation of these MRI sequences. Yeah, so they don't have like for classification or for any other injuries. Only the masks. Yeah. Okay. Yep. Yeah. I just want to.
Comment by this.
Bye. Je vais. C'est bon. Yeah. Yeah. Okay. I will go to the first processing step, right? Yep.
So if I could understand in this step, you would just select a fixed number of slices that could ignore the information that was not used for the network.
Exactly.
So my question here, do you think that it will be better to just apply a segmentation to detect the region of interest? You segment and then you feed the segmented area to the deep neural network for the classification.
Yes.
Why you have not done it? And do you think that this could improve the performance?
Yeah. Okay. So actually, this is done in the fourth contribution, in which we train. So instead of training a network to segment and then feed it to the classification, we want to train it into multitask network. So the network will do the segmentation of the knee joints of the area, and in the same network we share these weights with the classification pathway for the classification.
And this actually has improved the classification performance a lot. Yeah, this is, yeah. Indeed,
So the question, it would be better to
Yes.
invest.
Yeah.
Okay. You have chosen a lot of CNN-based architecture.
Yes.
And involve the contribution. So why CNN, and why you have not chosen like more recent architectures?
Yes. Yeah.
But have proved better performance for classification and segmentation.
Yeah. So this is indeed an excellent question. So why we haven't used VITs? So why we haven't used VIT or transformer or any yeah. So basically we kind of were restricted by both things. So the memory storage and the computational storage. So just to make sure that our architecture, our 3D CNS models and all the databases could not actually be trained on any different GPUs than our lab GPUs, because like we have three 140 gigabytes of GPUs that we train on multitask. So this is actually a very complex network to be trained on any network other than the CNS. So I'll just briefly mention like how, for example, for BIT. So just need to maybe hide this. Z is sleeping. So for example, for BIT, so the complexity of both, like the computational intensity and the memory storage and the memory need is quite quadratable or imaginable compared to only CNS. So for the patch for each MRI scans, like we have like let's say for this we have 100 slices because 3-dimensional, and we are going to extract the patches. So from each one MRI scan, we can extract up to 5,760 scans batches. So instead of training one network to only one MRI scan, we are going to train them like on 5,700 patches, in which this like the memory usage will expand, and also the computational intensity of using self-attention mechanism, because in the self-attention mechanism you are going to calculate the self-attention for this patch across to all 5,760 patches. So the complexity of training VIT, because we have tried it, and it was quite very complex only to train them, only very limited scans.
This is if we train it from scratch, but
Yeah.
for example, we can take a pre-trained V.
Exactly. Yeah.
And we just fine-tune it. We can just freeze even a few years and just we train in few years.
Yeah. So this is actually applicable also for CNS. But in our case, the data is 3-dimensional, and we don't have pre-trained model on 3-dimensional data. So all these CNNs or VIT is purely trained on ImageNet or any 2D data, but we cannot adapt it to 3-dimensional data directly. So what we did is, for example, for ImageNet models, like we converted all the network for 3-dimensional, but for VIT to train from scratch, it was purely like painful and really complex. Yeah. Yeah. Yeah.
I have a question about the multi-view fusion. Okay, I don't know in which slide you get.
We do multi-view basically for all network. Yeah.
If I remember, we have just concatenated the learned features from each view, and then you give it to classify. So my question here: why not apply a joint learning after the concatenation? Because usually it will give a better performance.
Okay. Yeah.
Then just what we call early fusion, because there is different category family of fusion. What we have proposed is what we call early fusion. So it will be better to do a model-based fusion.
Okay.
So it's to apply joint learning just after.
Okay. So it could be, for example, similar to this maybe. Like we combine them into one single neural network. And then we extract the features here, we do the classification.
After the class, okay, you will add another deep neural network. It could be Jennane, CNN. You can do any type of learning. Okay, and after that, last step, it will be this classifier to give the byte.
Okay, so I got you. So instead of just combining them here, we train a normal network
like to learn how to fuse the models, features from different modalities. It is, yeah. Mhm.
Maybe for future.
Yeah, yeah.
Or should
Yeah,
it will be
but-
for the purpose?
But for here, for example, here, we have studied like multiple fusion, like we studied first late fusion
Yeah.
and late fusion and early fusion. And I've seen that this late fusion is actually much
Yeah.
better.
Yes.
Yeah.
Yes. Yes.
Mhm.
Actually, there is early, late, embed, and
Yeah, yeah.
model based.
Yeah, yeah.
The model based is the okay. It will took much more of resources.
Yeah.
It will be more late,
Mhm.
but it will give a better performance.
Yeah.
Okay.
Sure, it's just.
And it will also give you better interpretation in the gram can.
Okay.
Okay. Actually, I like the idea of the M3 net. This idea of doing a multi-view, multi-label, multi-model.
Yeah.
But, however,
Yes.
how to have a technical contribution instead of using the 3D resonance. For me, to see this architecture and having, instead of having 3D resonance, something else that you put,
Yeah.
I don't know, like stochastic transformer,
Yeah, yeah.
like
Mhm.
another new
Yeah, yeah.
deep neural network, it could make like a huge contribution and
Yeah.
make like a big novelty in all the things.
Mhm.
Okay, so if you choose to redo all the work again, what you could modify here?
In the M3net?
Yeah.
This is interesting. Let's me. So basically, yes, for sure, like using different CNN models will surely maybe boost the performance and increase the performance.
CNN or other architecture?
Other architecture, like you've been like swing transformer or any transformer-based model. Yeah, it could be very, very efficient. Also, maybe some data implementation using GANs or additional neural network model to expand the number of scans and to have more holistic images rather than just the original images. Maybe this could be expand the or improve the classification performance. Yeah, and also maybe other injuries as well, like just not meniscus and cartilage, maybe the ASL and tendons and ligaments. Maybe this should be included into this neural network because we have their labels. Yeah.
Have like much more critical
Yes.
of your work today. I think that if you do the work again, you will be able to change this other stuffs.
M3net.
Or M3net or the rest.
So for the rest, sure. So for the first one, I don't have, to be honest. For this, maybe yes, different CNN models, different feature extractors, different neural networks. And for the last one, maybe I want to extend it in order to support like image reconstruction, MRI reconstruction, rather than just like the classification segmentation. I believe that you doing a reconstruction with segmentation will extract more important features rather than just segmentation. So maybe that could lead to better performance. Yes, I have a conference paper and one journal paper under review. Yeah.
But I think instance from slices.
Yes. Okay. So this is it.
Yes, yes.
So here is just so this is purely into the neural network. So this is not as a pre-processing step. So we put the MRI scan into the neural network, and into the neural network we are going to loop over all the slices that exist because we can consider it as a NumPy array, multi-dimensional NumPy array.
Okay.
We are going to loop over all the dimensions of this NumPy array, and then we are going to extract them. And then we are going to, the moment we are extracting them, so here, as like we are have a batch, like batch of 16 or batch of 17,
Yes.
then we put all these batch into the neural network. So this is like a tricky way on how to trick the neural network to us as input 3D MRI, but we train this MRI using a 2D CNN.
Here there is no feature extract.
No, no.
Just will concatenate the information from the slices in different ways to get different features.
So this is the feature extraction actually exists.
Yes, this part we got it, but
Yeah.
the first.
But here, no, there's no feature extraction, just slice extraction.
Maybe, yeah.
Okay. It's like you reorganize the 3D slices into
To the plane.
to the plane, but it will be like a huge.
To the plane, just imagine it as 3D plane. But how huge, for example?
In terms of size.
The batch actually, if you train not network, the batch size is 1.
No. That's the whole plane.
Yes. Yeah, for example, each MRI slice, for example, this corner has like 70 slices. So yes, so 70, I think it's okay for a batch size.
Multiplied by 250, I don't know what's the.
Oh, yes. Yeah, it's gonna be very, very high, yes. But it's purely two-dimensional. Like this is, like these images are two-dimensional, like not three-dimensional.
Yeah.
After?
Do you have any problem with CPU or GPU when you train this model?
It was quite lightweight compared to purely when we don't do like 3D.
So what's the size of this huge block that you feed to the res squeeze net?
What do you mean like the size?
Here it's
the number of slices. So it's going to be like, for example, 380 by 70 slices. By 70 images. Yeah, it's huge, quite huge. Yes. And that's why I told you, like, our experiments could not be conducted without our lab GPU. Yeah.
So the idea of previously not using a bit is
Yes.
maybe
Yeah.
you could have adapted about it.
Yes.
Trinity this.
Yeah.
Maybe bit it will be visible. Yes.
Mhm.
Okay. So the risk squeeze net, is it yours? Your proposed network or you?
Yes. No, it's out. Yeah.
So you have a publication and you submit something?
Mhm.
Okay.
Yeah, one conference paper and one journal paper.
Okay, good. I think the last question for me, it will be in the multitask approach. Did you train your networks simultaneously for the two tasks or it should branch separately?
Yeah. So this is the multitask network.
Yes.
So it is an encoder we call the network, and from the encoder we extract classification pathway. So it is into one single network. So this is like not two branches or two different CNNs or two different encoder decoder. So this is purely one network that does the classification and segmentation.
So you train it simultaneously for
Yes.
classification
And
and the segmentation.
Yes.
Okay.
Yeah.
Okay. Have you think about like adding some contraceptive loss
Yes.
between the two tasks to improve
Yes.
the classification and the segmentation?
Yes. This is actually in our mind is like to employ contrastive loss or any other losses like to do that. But this is purely good idea, yeah. And this is the last contribution that we conducted, and we couldn't complete it because we have so many also ideas to do. This is, for example, idea that we want to implement. Sorry. So this is the. Sorry, this is not. I don't have the. This is it. So actually, the network will be much complicated and much better than just using segmentation or classification. We want also to do the reconstruction. So the reconstruction, we are going to extract more important features than just the classification, the segmentation masks.
Think about for the next part, if you could add like some contraceptive loss between the two, it could improve the results and the performance of the two branches.
Okay.
So thank you.
Thank you so much for the questions.
Merci Alice pour cette question. Je passe maintenant la parole à ADEL Mouloud, Université d'Aix-en-Provence, qui est le deuxième second rapporteur.
Merci, Président. Thanks very much, dear Chair of the panel. First of all, I want just to thank Rachid for inviting me to be a member of this panel. I want also to thank you for the huge amount of work you've done in this PhD.
Thank you so much.
And the very clear, comprehensive, and defense and presentation of your work. So I have just a lot of questions have been asked by my colleagues. I'm going to squeeze them. And then just ask you some more general questions and perhaps one or two technical ones about your contributions.
Yeah.
Okay. So it was my pleasure to read your PhD manuscript, but you know, we can congratulate, but we need also to give some comments. I think that, yeah, in your, the way you have written your manuscript, you could perhaps do some of less redundancy. That means that a lot of things have been appear in every chapter, more general things about, for example, the fact that the MRI is more suitable for soft tissue than the X-rays, something like that. We have found them in already every chapter. I think it would be interesting for you, for the manuscript, just to make it more efficient in terms of summarizing it a little bit more in these aspects. This is the first thing I wanted to tell you. The second thing is a more general comment. I have not perhaps found any aspects on your PhD manuscript about your collaboration with the clinical team, with doctors. Was it the case or not? Or perhaps I have something, I don't know.
Yeah. Actually, we didn't have any contribution with any medical experts during my PhD. Only some just separate emails like from doctors that I don't know with, and just contribute with them. But yeah, the aspect of contributing with any medical experts was kind of missing. Yeah.
Because it's important for you as a applied mathematics scientist just to show results to doctors, not just to have their.
Yeah. Maybe I missed your question. So basically, this is part of ADEL Mimosa project. So this is just like between University of Orléans and Mame, which is a company in Switzerland. So they are like a company that are focusing on making our models into production. So my work is like to do research, to do validation of my work, and also to make these models into production. And this actually, the enterprise will be in direct contact with the medical doctors and with the radiologists and everything. So this is purely from their end.
A more general question about these first questions is when looking to the tables you have put on your manuscript, you have the accuracy that appears, but I have never seen the specificity, sensitivity. Why? Is it a choice or?
Yeah, actually, so our manuscript has been written solely from our research papers, and we consolidate them into the manuscripts. So each part is part of journal or conference plus journal paper. So this why we include only this matrix because like we want to compare our results into the state of the art, and state of the art only uses the AUC value. So we rarely see the accuracy or the specificity of the precision. But we have like these values, but I forgot to put them in the manuscripts. But yeah, this is purely for comparison perspective with state-of-the-art models.
Okay, thank you. So for these general aspects, then perhaps go into some technical questions. So for the first contribution, okay, the 3D RESI and GAP, what is the significance of incorporating this global average, putting a number of this architecture, Wi-Fi? Is it?
Yes. Okay, so yeah. So this is purely just to like very first question answer is that it's purely experimental. But however, I will just first mention the idea of like concatenating global average pooling. So the idea here was we have seen that after analyzing like without global average pooling, so we have seen that there are multiple local features that are extracted into separate convolution blocks that are kind of lost or are not preserved. So how to preserve them is that we want to make them as a separate feature vector, and to do that, I want to summarize them into the global average pooling.
and then we concatenate all of them at the end of the neural network. So this is purely like sequential convolution features, and this is like concatenation of local extracted features from 3D convolution logs. Also, we did some data ablation. If you want to interested, like to see the effectiveness of removing this, and you have seen that yes, it's quite important to include gap models. 1 second. So yeah. So here in the ablation study, so like we take the gap and visual connections. So this is purely like business. Let's see. And like what we include, it does included this and included the identity blocks. So we mask the global average pooling concatenation. And we have seen that the classification result will reduce. Also, we have like hidden and also masked the residual connection, remove them, and also the classification result will reduce as well. So by adding them back, the overall classification performance will boost again to up to 93.2%.
So second technical question about your second contribution. You have a process of fusion, yeah. And then so you compare the different ways of doing fusion, linear one and so on. But just you mentioned that it's a linear one, but it, to my opinion, it's just a particular case of linear ones because you are weighting by exactly the same way in different components. Why didn't you do something with differently weighting the different components?
Actually, it's not just linear, like I've seen that it's concatenation, linear, and averaging, and multimodal attention infusion. But in the linear,
in the linear one, you have just added.
Just added the features.
You weight them.
What do you mean by weight them? Like weight the features based
exactly. Weight them with some parameters. Instead of
this is interesting.
Giving just one. You are just weighting the components by one parameters, and the parameter is 1. You know. Why did you? What do you think about weighting them, giving more importance to some components than the others?
Okay, so basically this was done like using the multimodal attention fusion. So the multimodal attention fusion was done is that it will take the linear fusion or the concatenation, like we can set them, and it will learn which features are more representative and will give them more weights. However, it turns out like doing only simple linear fusion is much better than using attention fusion, linear fusion. So this was quite surprising, but we didn't dig into it to see why quite difference between the multimodal attention. Even it will give the important features more weights when it's going to combine them. Yeah, but it's quite interesting, yeah.
So, in the MVMI.
MVMI.
What might the introduction of segmentation map, as mentioned, for the following chart to enhance the model classification system?
You mean this slide?
I have another slide, but it's a general question about your contribution. I want just to, if we introduce some segmentation map, does it help your model to be more improved or not?
Yes, certainly. And this is what we did in the final contribution. So this is like we include like both the MRI scan and the segmentation map. Like and this actually improved the results like much better than just cropping them. Yeah, certainly yes.
And about using Euro 8. Euro, we know Euro is a well-known architecture for object projection and so on, but it has been trained on more natural images. Can you mention that? So how did you use it? Did you use a pre-trained models? Did you use it using your own images?
Yes.
What did you do?
Yes, very nice question. So YOLO V8, as you have mentioned, is well-known architecture, like employed in computer vision, etc. However, its point, as you have mentioned, is quite limited to only like very nature objects. So here, what we do is that we take the YOLO V8 model, which was that state of the art at the time of writing this paper, because now we are in YOLO V12 or 11. So we take the weights of YOLO V8 and we fine-tune them on MRI data. So YOLO V8 was actually trained like they give them objects and they annotate them like bounding boxes. So what we do here is that we provide bounding boxes for the knee MRI slices, and then we train your V8 model like again and like fine tune it. Yeah.
Question. In the floor to my colleagues. About future dimensions and future work.
Yeah.
So, what do you about, to your opinion, or to your, what is the most important? You have mentioned a lot of
future work.
ways of improving your work and so on.
Yeah.
What is the most important? If we do a hierarchy about things that would be of interest or that would be make your contribution more developed, and how can you improve this? And is there any if you
Any order?
rank them?
Yes.
What would be according to you the most important thing you have to work on?
Exactly. This is a very nice question. And first thing that I personally we want to do it is like to continue the complete the fast final contribution. This is the first one because like we have the models and everything. But interestingly, like for future directions, is that the first. This is the one thing that I would like to explore is to use what we call vision language models and segment anything model or medical segment anything model. This is what will allow us not just to segment the mask of the knee joint, but rather than also to segment different components of the knee joint, like the femur, the cartilage, the meniscus, the tibia, etc. And I believe this will expand and boost the product classification performance. Yeah, for sure. Additionally, like for many reviews papers that we have done, we have received, is that the diverse generalizability of the models. Like we want to maybe train our models maybe on different data sets, but we need some ground truth labels. But yeah, maybe the idea of you that you mentioned collaboration with any medical experts to do ground truth labels for us would potentially help us in this field. Yeah.
Thank you once again. Congratulations.
Thank you so much.
On what you have done.
Thank you so much.
These shares. I'm happy for my questions.
Thank you. Bon, je vais donner la parole à un peu plus loin et ensuite on reviendra vers les micros. Dr. Mohamed JARRAYA, if you want to ask questions, so the floor is yours. I don't know if you can hear us well or not.
I can hear you. I had to put, thank you so much. I had to put a headphones because I don't know if it's luck or bad of luck, but they just started construction behind me. So now with the headphones, hopefully I can hear you better. Anyhow, Mohamed.
Due to Z or not?
Comment? How was that?
Due to Z?
No. Z is very quiet. Z is very quiet, at least today. But the construction is like on the street and behind. But Mohamed, this is a very, very nice work. Really, like I congratulate you for this and your supervisor. This is really fantastic. Some of the questions that I wanted to ask have already been asked, so I'm going to be brief. But if you go to contribution 1 and your slide about results, with that table, with the single view versus multi-view, yes, right there. So I have a question here for you is, in your opinion here, what do you think, why do you think that the coronal reformats, the ACC and the AUC seem to be higher for the coronal reformat compared to the other two sequences, the sagittal 3D DES and the IW?
Yeah.
Do you have any hypothesis on that?
Yeah, I have like purely medical hypothesis, not computational hypothesis, on why the classification performance using coronal MPR is much better than the other acquisitions. So coronal MPR is coronal reformat acquisition, which is like was extracted and reformatted or reconstructed from sagittal disc to form a new holistic view of the knee MRI from the coronal side. And this actually in the coronal view, we can see more details, like the cartilage compared to sagittal from the sagittal view. And I believe that was what makes the feature extraction from coronal MPR or reformat much better compared to other sequences. Additionally to this, the number of slices, for example, if we compare coronal MPR, was quite around 60 slices per scan, and this sagittal disc was like 160 slices. So the computational load and the complexity of sagittal disc will be much complex compared to coronal reformat.
Okay. Yeah. Yeah. I think your hypothesis is quite reasonable here. Like, you know, the coronal reformat, after all, provide a view that's kind of similar to the X-ray, but then it has a higher resolution because it's a 3D. And so, yeah, maybe like the osteophytes are better seen, which helps with the classification task. The other thing, Mohamed, is so here you're heavily sort of relying on the DES. The reformat is reconstructed out. So the coronal reformat is reconstructed out of the DES, right? So you're kind of, the contrast resolution is basically the same. It's only the view that's different between these two sequences. But what I wanted to ask is that, so to what extent do you think the reliance on specific MRI sequences like the 3D DES basically will impact the model's performance and generalizability here?
Okay, I think I got your question. So basically here, yeah, indeed. So the coronal reformat was kind of extracted from sagittal disc. So we kind of, maybe we could, so basically like the construction, the pixels and everything will kind of be the same for the reformat. However, here we can see the features from the sagittal view that cannot be seen, and structures from the sagittal view that cannot be seen or interpreted from the coronal view. So it's like, for example, having an image that you can see it. I actually have brought one example that might be much better for the audience to understand. So for example, instead like of showing like this is a car. So this is a car, so this is the image of the car, but if this is also the image of the car, this is also the image of the car, but also the image of the car. Actually, if we merge all these views, we will have a more holistic view. And additionally, yes, the pixels of the sagittal and the coronal will be quite the same. However, the features and that we can see, or the structures or the components that we can see from here are much different from other views. And this actually applies to MRI, because I don't understand much about MRI, but here as a car, I can explain it better. Yeah, I hope I got your question and I answered it.
Yes, yeah, that's fine. No, the last thing is like well, you know, the last thing is like a purely clinical question. So I'm not, you know, I'm not gonna expect you to have like really an answer, but like this is really a food for thought. But so for your you use AUC and ACC for you know, these computer vision metrics basically that you're using to assess your model. Do you think these are the most relevant clinically at the end? You know, is that what makes your models clinically relevant, basically?
Yeah, I think.
And again, yeah, go ahead.
I think in clinical practice, what you usually use, but we don't usually use, what you usually use is the sensitivity, right? I think that would be the most reasonable matrix for you to use in terms of assessing if the model much performance and that we can rely on into production or not. Not sure it is the right matrix that you use. Yeah,
that's fine. Yeah, that's these are the few questions that I had, but this is very nice work.
Thank you, Mohamed. On va passer maintenant plus en local. Je vais passer peut-être la parole tout d'abord à Diarra. Si tu veux poser des questions.
Oui, alors oui, je sais pas la français.
Pas grave.
Déjà, je voulais te remercier pour ton travail. J'ai pris un énorme plaisir à lire ton manuscrit. Et voilà, il s'agit d'un très joli travail et je suis impressionnée par la qualité des résultats. Donc, voilà, je trouve que ça avance en tout cas pas mal la recherche sur ça et ça ouvre pas mal de perspectives aussi. Et personnellement, ça m'a donné aussi des idées. Donc, merci. Merci, félicitations et puis merci à Rachid aussi pour l'invitation. Alors, j'ai juste une première remarque avant des questions. Ce qui m'a manqué un tout petit peu dans le manuscrit, c'est qu'il me manquait comment dire, des informations sur l'entraînement de tes modèles, la complexité. Il y a des modèles, donc je doute que certains sont plus complexes que d'autres. Et je n'avais pas ces informations-là sur voilà, les temps de calcul, d'entraînement des différents réseaux, etc. Donc, ça m'a un peu manqué. Et après là.
I can have the answer if you want. J'ai la réponse si vous voulez la réponse. En fait, c'était la dernière expérimentation ou bien la dernière contribution où on a pris attention, on n'a pas mentionné la complexité des modèles parce que c'est un aspect très important qu'on avait déjà mentionné. En fait, là, on compare la taille et la complexité d'entraînement du modèle en termes de taille, en grosso modo de détail. Mais aussi, on peut, par exemple, introduire d'autres métriques, par exemple, quand la complexité d'entraînement, par exemple, le temps qu'il prend pour entraîner et tout. C'est un aspect très important. Ok, je vais noter ça.
J'ai quelques petites questions. Alors, tu en as pas parlé ici, mais pour ton modèle PhD sur les sauts de la bête, parce que on voit que ça ajoute quand même sur les performances. Et comment tu choisis, par exemple, combien d'images non annotées tu choisis, etc. Donc, est-ce que tu avais un critère sur ça ou comment tu les as choisis?
Ok, pour ça, je vais expliquer en anglais parce que je serai plus à l'aise. D'accord, merci. So the first step is that we train this model and we make sure that it's really powerful and really optimized. So there is no overfitting issue, there is no just not hallucination, but after that we make prediction on 250 scans. So the initial label data set has 900 scans for 450 and another 450. So the database is quite balanced. So the model will likely not be biased towards one class or to another. And the reason that we take 250 scans is that we cannot take like 2,000, 1,000 to predict them with the model directly. Is that we need to take few numbers of scans. Like here, we take less than a third of the number of scans, and we make prediction. However, how we choose the scans labels. So here, we set a range here, and to consider that the image is going to be considered as positive, or it's going to be considered as negative. So based on the output probability, so the output probability is going to be from 0 to 1, and if it's close to 0, like from 0.4 to 1 to 0, it's going to be considered as negative. And we did some uncertainty level, for example, for scans in which the model, like say, this is 0.5, like 50%, I'm not sure, it's like this or like this. So we don't consider these scans to be included here. So we remove them, we set some certainty level. Yeah.
Je regarde tes résultats, justement, page 33, ça prononce un peu la question de Mohamed. Et j'ai l'impression que la vue 3 n'apporte pas beaucoup d'informations en multiview. Est-ce que je me trompe ou pas? Parce que quand je regarde toutes les performances entre 1 plus 3 et 1 plus 2 plus 3, j'ai l'impression que la vue 3 n'apporte pas beaucoup plus de performance en tout cas.
Beaucoup plus de performance en considérant seulement cette acquisition, c'est ça?
En multiview, là.
En multiview,
est-ce que 1 plus 3 en gros ne suffisent pas? Est-ce que
2 plus 3.
1 plus 3 au lieu de 1 plus 2 plus 3?
Oui, ça rajoute 1 plus quelque chose, 1 plus 2%, mais c'est déjà quelques dizaines de scans déjà. Donc, ça a rapport un peu d'accuracy. Par exemple, on a dit, par exemple, 4000 scans.
Justement, c'est ça que je me demande,
Ouais.
par exemple, au coût, parce que c'est toujours comment trouver le bon équilibre entre ce que ça te coûte et ce que ça t'apporte. Voilà, donc, comme je n'ai pas l'info sur est-ce que, en termes comment dire, en termes computationnels, ça ajoute plus de complexité, forcément, quand tu rajoutes là. Est-ce que ça t'apporte tout? Est-ce que ça t'apporte gagner un peu de performance?
Oui, je suis totalement d'accord. Parce que ça vaut le coup de concaténer, par exemple, du droit, alors que, par exemple, ça sera suffisant.
Oui, voilà. Ouais.
Mais en termes, par exemple, ça, par exemple, 1 plus 2, 1 plus 4, c'est à peu près de 50 scans. 50 scans qui ont été accurately classified. Je pense que c'est oui. Pas beaucoup. Ouais.
Ok, d'accord. Et dernière question, mais elle est assez générale, ma question. Ouais. Est-ce que tu penses que les méthodes que tu as développées donc pour l'arthrose du genou sont facilement transférables pour étudier d'autres articulations que le genou? Et quelles seraient les adaptations à apporter si on voulait les appliquer à du X-ray, par exemple?
Ok, je réponds pour la première question d'abord. Donc, on a étudié pas que l'arthrose de genou dans cette thèse, on a étudié le méniscus aussi, le cartilage et aussi l'arthrose. Je pense que nos modèles sont performants dans tout ce qui concerne le knee MRI. Donc, tout ce qui concerne l'extraction de features de knee MRI, la classification, la prédiction, aussi la segmentation de la chapitre. Je pense que oui, mais ça reste toujours à vérifier, peut-être. Et pour la deuxième question, c'était pour l'adaptation. OK, pour l'adaptation, donc nos modèles sont purement 3D. Purement 3D, donc je ne pense pas que ça va être facilement adapté en 2D.
Du 3D, mais avec
2D. Voilà. Mais ça reste toujours, ouais. Oui, mais ça reste toujours que le modèle pré-entraîné là-bas, oui, pré-entraîné, ça reste toujours, il reçoit comme un input des données quand ils sont 3D. Donc, ça, c'est un critère très important. Je pense pas, personnellement pas. Ouais, je pense pas. Ouais. Ouais.
Parce que je t'appelle que quand j'ai vu le dernier modèle, je me suis dit, OK, donc en fait, et en plus, quand je dis, ouais, donc tout ce qui était avant, voilà. On regarde que le dernier modèle. Assez...
Par contre, par exemple, nos modèles, par exemple, ils ne sont pas vraiment biaisés, par exemple, et ça, c'est une performance seulement pour les coronales, ou bien pour le sagittal, ou bien l'axial. Nos modèles pour l'IRM sont performants dans toutes les vues, dans toutes les acquisitions. Par contre, pour d'autres types d'imagerie, que ce soit par exemple X-ray ou d'autres, je ne sais pas. Ouais.
En tout cas, merci.
Je vous en prie. Merci beaucoup.
Merci, Diarra. Je vais donner la parole maintenant à Sylvie Treuillet, qui est à l'Université d'Orléans.
Merci, Aladine. Je vais rester en français.
Non, c'est pas grave. Ouais.
Je voulais remercier aussi Rachid, bien sûr, de m'avoir invité. J'ai beaucoup apprécié et la substance et le manuscrit écrit. Je vois mon collègue sur quelques, effectivement, redondances. Je pense que c'est parce que les chapitres ont peut-être été aussi articulés comme les articles.
Ouais.
Et souvent, ça pêche.
Ouais.
Peut-être que j'ai manqué des choses, mais il y a des choses que je n'ai pas tout à fait bien comprises. Et donc, je vais essayer d'éclaircir ça avec vous. Notamment, je vais revenir moi, tout au début sur le pré-traitement des données, en fait. Oui. Et sur ce que vous donnez en entrée de vos réseaux. Donc, j'ai compris qu'on partait d'un volume 3D.
Ouais.
J'ai mon émiré qui me dit à la tête. Et du coup, donc, vous sélectionnez une sous-zone, ce qui paraît logique, on ne va pas prendre toute l'image. Et ensuite, vous sélectionnez certaines, si j'ai bien compris, certaines slides à l'intérieur de ce volume, également pour vous rapprocher des informations les plus pertinentes.
Exactement.
Combien, j'ai absolument bien compris, c'est combien de slides vous donnez en entrée pour chaque vue de du premier réseau?
D'accord. D'accord. En fait, je n'ai pas la réponse ici sur le slide, mais je l'ai sur le manuscrit. En fait, ça dépend de chaque acquisition, ça dépend de chaque vue. Par exemple, dans l'acquisition sagittal desk, par exemple, juste, on va tout bas. Ici, par exemple, pour celle-là, par exemple, on a 160 slices. On a dû supprimer 30 slices de ces 160. Pour celle-là et celle-là, on a supprimé à peu près 10 à 15 slices. Donc, ça dépend du séquence et ça dépend du vue.
En tout cas, il y en a même un grand nombre.
Exactement ça, oui, c'est exactement.
Et cette suppression, bon, elle a été faite alors, avec un expert médical, sans expert médical, ou on est...
Au début, c'était comment? Donc, on a deux façons pour le pré-processing. On a cette méthode qui est vraiment semi-automatique. Donc, au début, on a vu que sont les slices qui sont vraiment pas pertinents. Les slices qui sont pas pertinents, c'était vraiment très clair qu'il n'y a pas d'information pour le joint d'articulation. Et du coup, ça, c'est pas vraiment pratique. Parce que pas toutes les slices, pas toutes les scans ont les mêmes nombres de slices. Et aussi, la rotation, il y a des fois la rotation du genou, ça, des fois, pour quelques scans, ça va être ici, d'autres, ça va être ici. Donc, le cropping aussi, ça a un impact. Donc, pour cela, ça nous amène pour le deuxième pré-processing qu'on a pré-entraîné YOLO. Donc, YOLO, c'est un peu parfait, mais ça détecte quels sont les slices qui sont à relever et quelle est la région à détecter. Okay. Donc, quel que soit au début ou au milieu.
Donc, au départ, vous avez commencé manuellement, et puis ensuite, vous avez entraîné YOLO pour le faire. Et donc, du coup, le format, en fait, donc la résolution de chaque boîte, de chaque ligne d'intérêt est la même pour tous les modèles.
Exactement. Donc, c'est une excellente remarque. Et je dois juste balayer. Ouais. Oui, oui. En fait, on a un état post-processing. Donc, à force que toutes les slices ou bien toutes les scans n'ont pas les mêmes paramètres, et aussi l'emplacement du genou, l'articulation du genou, ce n'est pas pour toutes les slices. Donc, forcément, qu'un cropping, ça va être par exemple 120 pixels pour 160, et pour un autre slice, par exemple, ça sera par exemple comme ça. Par exemple, celle-là c'est grand, celle-là c'est petit. Donc, on a ici un état de post-processing où on va mettre toutes les slices dans le même scale. Ouais.
Un scaling.
Un scaling, ouais. Ouais.
Très bien. Très bien, ça répond bien. Donc, en fait, en entrée, si je synthétise, en entrée de chacune de vos vues, vous n'avez pas forcément le nombre de slides.
Non.
Très bien. Maintenant, quand vous vous comparez à l'état de l'art, donc il y a un certain nombre de tableaux sur lesquels on a des pourcentages. Est-ce que ces chiffres sont sur des bases de données exactement identiques?
Donc, en fait, pour tout ça, par exemple, pour la première contribution, on a dû comparer avec plein de travail, que ce soit sur la même base ou pas la même base. Parce que au début de ma thèse, on n'a pas trouvé beaucoup de papiers sur l'arthrose de genou en utilisant les IRM, en utilisant le white database. Donc, c'était vraiment restreint, mais on a dit on va juste comparer avec n'importe quelle base, n'importe quel essentiel, qu'elle soit en IRM. Par contre, pour les autres contributions, on a comparé que le papier utilise la même base et utilise aussi les mêmes labels que nous, on utilise.
Vous avez réentraîné les mêmes
non, on a trouvé quelques papiers qui utilisent les mêmes bases et les mêmes scans. Donc, on a comparé avec eux directement.
Et comme tu utilises une sous-partie de la base, tu n'es pas sûr que ce soit exactement les mêmes scans qui aient été sélectionnés.
Ouais.
Je me trompe, ça, je dis ça. C'est pas forcément grave, mais en tout cas, pour une présentation, on va dire, parfaitement honnête, d'abord, je ne pense pas que le tableau de droite soit dans la thèse. Je le trouve assez intéressant. C'est le nom. C'est moi qui ai été. Parce que quand je l'ai vu, j'ai trouvé ça très bien. Et ensuite, voilà, il faudrait le dire moins dans les commentaires. Parce que du coup, on compare quand même des choses qui ne sont pas complètement comparables. Ça ne changera peut-être pas grand-chose au pourcentage, encore que Encore que pour avoir fait des essais dans d'autres types, même quand on fait par exemple, la question a été posée sur la généralisation des bases, on est parfois étonné quand on teste des moulins entraînés sur une base, quand on les teste sur une autre base, alors que la seconde base paraît très très proche, avec des images que nous à l'œil on ne voit pas différentes de la première base. Des fois les résultats s'écroulent relativement. Hein. Donc c'est voilà, il faut toujours être un petit peu prudent, je pense, par rapport à cette question sur la génération. Et d'ailleurs, dans ce sens-là, la question n'a pas été encore posée, au final, combien de patients, combien de scans représentent la partie test sur laquelle tu calcules ces chiffres
Ça dépend de quelle expérimentation, de quelle contribution.
Alors, par exemple, sur le premier modèle.
Pour le premier modèle, on prend à 450, ouais. On prend 30% pour 20% pour le test et 10% pour la validation. Ouais.
On teste sur 20% de
oui.
450.
Ouais.
Donc les questions une question supplémentaire sur l'apprentissage semi-supervisé. Donc, pour propager tes pseudo-labels, tu utilisé un seuil.
Oui.
Pour savoir ce que tu gardes ou pas. Si j'ai bien vu dans la thèse, donc le seuil est à 50%. Ouais. C'est ça. Le choix de ce seuil, 50%, c'est ce qui est communément fait par tout le monde. Est-ce qu'on a essayé plusieurs?
En fait, ça c'est une erreur de frappe. Mais ce n'est pas une erreur de frappe. On a fait ça, mais on a dû refaire ça. Au début, on a fait le seuil de 50%, mais à la base de tester encore, on a trouvé qu'il faut mieux faire une certaine seuil. C'est ça que j'avais répondu à Mme Diarra. Oui. C'est qu'on a fait une seuil de certainité. Par exemple, entre 0,4 et 0,7, le modèle là n'est pas certain. Donc, par exemple, sur 50%, il n'est pas vraiment certain que ce sera 0 ou 1. Donc, on n'utilise pas, on ne prend pas ces scans-là. Mais pour l'autre, par exemple, de 0 jusqu'à 0,4 et de 0,7 jusqu'à 1, on prend comme négatif ou positif.
Oui. Je trouve que moi, quand j'ai vu, j'ai trouvé que 0,5, c'était.
Ouais, c'était pas mal. Ouais, c'est exactement ça.
Après, on alimente ensuite le modèle avec ça. Alors, est-ce qu'on ne va pas lui donner trop de confusion? Et l'autre chose aussi, bon, tu l'as un petit peu évoqué, que je trouve qui aurait été intéressant, c'est d'avoir. Tu fais combien d'itérations au total? Et justement, le nombre d'itérations, il va jouer. Sans doute, à partir de ce seuil, là, ça aurait été intéressant de voir ça.
C'est exactement ça. Donc, au début, par exemple, on a 4700 patients. Pour chaque patient, on a des scans d'un genou ou deux genoux. Donc, au total, on a à peu près 9000 scans, 9700 au total. Mais du coup, on a été limité par le nombre de scans qu'on peut utiliser pour entraîner nos modèles. Donc, au total, on a pu, par exemple, pour cette acquisition-là, on a pu labelliser à peu près de 3400, 3300, ça dépend d'acquisition. Mais à certains moments, on a dû, il faut arrêter parce que même si on continue à labelliser, on entraîne plus, ça ne va pas être suffisant pour notre GPU à entraîner plus que ça. Par contre, par exemple, le nombre total des scans, c'était 12 000 scans. Donc 12 000 multi-vues, donc à peu près 3 000 sagittal d'Est, 4 000 coronal et 4 000 sagittal TSE. Donc à peu près la combinaison dans un seul modèle pour multi-vues, c'était vraiment challenging.
Oui, bon voilà, c'est pareil, hein, les pourcentages, il faut se méfier, le nombre de scans, enfin plus tu as un ensemble d'entraînements important, plus naturellement, quel que soit, même sans contribution.
Oui, c'est exactement ça.
Voilà. Donc, il faut méfier un peu de ça. Ouais, j'ai des plein de questions, mais c'est vrai que le temps passe. Je vais, sur la partie multitâche, j'avoue que c'est la partie qui m'a, moi, par rapport aussi à des questions qui ont déjà été posées, je me suis vraiment posé la question du pourquoi. Pourquoi est-ce qu'on a besoin de donner à la fois le diagnostic et la segmentation? Dans la discussion qu'il y a eu avant, en même temps, je comprends que la segmentation, ça serait intéressant plutôt de la mettre en amont pour pouvoir guider, justement, forcer l'attention du réseau sur certaines zones. Autant faire un réseau qui donne à la fois. Est-ce que, d'un point de vue diagnostic, ça a un intérêt de donner une sortie à la fois sur la segmentation et sur le diagnostic?
Je pense que oui. Pour un point de vue diagnostic, par exemple, peut-être Dr Mohamed BERRIMI va nous confirmer ça, mais en point de vue computer science ou deep learning, ça joue un rôle très important. Donc, au lieu de, par exemple, faire un simple cramping, par exemple, pourquoi pas on nous donne au réseau de neurones les régions plus précises. Par exemple, voici la région plus précise et qui contient les features plus importantes. Et ça joue un rôle très important. Donc, il y a quelques recherches qui utilisent, par exemple, à la fois, par exemple, on va faire la segmentation et on va extraire les segmented maps. Après, on va utiliser ces segmented maps pour la classification. Mais nous, ce qu'on a fait, c'est qu'on a fait tout ça dans un modèle multitâche. Et c'était vraiment impressionnant pour les résultats.
Mais ce que je n'ai pas compris dans le modèle multitâche, tu réutilises la segmentation pour la classification. Moi, je n'ai pas vu ça. Non, c'est en perspective.
Oui, peut-être. Ouais, mais pour ça, on utilise par exemple le même modèle, mais on extrait des features qui ont été extraites par le décodeur et l'encodeur pour la segmentation, et on va les utiliser pour la classification. Parce que, ouais.
Oui, il y a beaucoup de travaux qui injectent, par exemple, une carte de distance qui est faite à partir de la segmentation pour justement pondérer, en fait, l'extraction des primitives, là où il y a les zones les plus importantes liées à la segmentation. C'est beaucoup utilisé comme en imagerie cardiaque, émérique cardiaque aussi, par exemple. Donc, on peut le faire. Il y a aussi quelques travaux qui utilisent la segmentation comme un phénomène d'attention, en fait, à l'intérieur des réseaux. C'est fait aussi. Et alors, je vais revenir sur les histoires de diagnostic avec une question un peu plus générale, donc qui a été un petit peu déjà évoquée par rapport aux métriques qui ont été données. C'est une question qui va être embêtée, mais il faut la prendre comme une question un peu perspective. Les résultats sont impressionnants en quantité, mais aussi en qualité. On atteint maintenant des pourcentages de 93 et quelques %. Alors, est-ce que d'un point de vue médical, avec un tel pourcentage, on peut estimer que le problème est résolu? Je vais dire la question autrement.
Oui.
Est-ce qu'on atteint des... Voilà, déjà pour la question, c'est suffisant. Et puis l'autre chose, est-ce que si on se compare à ce que font les experts, soit en termes de pourcentage d'erreurs intra ou inter-experts médicaux, est-ce que là, maintenant, voilà, on est arrivé au bout, quoi, hein? Est-ce qu'on peut continuer à gratter des pourcentages? C'est bizarre comme question.
Oui, c'est dur, mais c'est une question plutôt philosophique. Est-ce que, par exemple, si on arrive à un certain... Par exemple, si on arrive à une classification 99,99, que ça a été validé dans plusieurs phases, est-ce qu'on va dire que ce domaine-là a été résolu ou non? Ça reste toujours un point de vue, peut-être, je pense, reste à confirmer avec les médecins, avec des experts. On va dire, par exemple, là, le modèle ne va pas vraiment faire attention à certains seuils. Je ne sais pas. Ça reste à valider avec les experts. Ouais.
Merci pour ta réponse. Ce n'est pas philosophique, hein, c'est une réponse très concrète. C'est-à-dire, en fait, il faut comparer quelle est la précision statistique entre les experts ou le domaine expert, donc ce qu'on appelle le intra-expert, les intra-experts. Le même expert, si tu lui montres plusieurs fois la même radio, il a lui-même une erreur. Tout ça, c'est estimé. Si maintenant on a des réseaux, peut-être que ça ne sert à rien d'être à 99%, peut-être que 90 ou même 92, ça suffit en termes de diagnostic. Je pense que la faiblesse de toutes ces approches pour l'instant, on l'a dit au début, c'est plutôt la généralisation. C'est là. Je pense qu'on a de très beaux résultats. Reste à voir si c'est valable sur des très grandes bases de données de tests, si les résultats ne s'écroulent pas, parce que c'est souvent le cas statistiquement. Parce que là, on est sur 20% de 450 cas, donc ce n'est pas énorme par rapport à la variabilité des êtres humains, etc. En tout cas, c'est surtout dans le cadre d'un projet ANR et à vocation médicale pour le diagnostic. Tout ce travail-là, il doit être fait pour le diagnostic, pour revenir à... Pour le diagnostic. Alors, je vais terminer sur le...
Merci. Merci Sylvie. Peut-être que je vais prendre la parole avant de te laisser conclure, Rachid. Merci Mohamed pour la présentation que j'ai appréciée, qui était claire, qui reprenait contribution et qui a essayé un petit peu de prendre des limitations des contributions précédentes et de les reprendre ici pour justifier la méthodologie proposée ou les méthodologies proposées. Alors, moi, j'ai quelques questions. Certaines ont été déjà posées, c'est d'habitude. J'ai passé le dernier, on passe beaucoup de choix, mais bon, on va essayer de trouver quelques questions. Moi, j'ai une question sur le 3DRS TNN gap.
Ouais.
Deux questions là-dessus, en fait. Dans le manuscrit, j'ai vu que tu as mis en tableau que tu avais mis gap et résidual.
Ah, pour
Tu as fait
l'ablation.
l'ablation study. Mais tu n'as pas fait l'ablation study du gap sur différents niveaux,
Ah non,
si je ne me trompe pas.
je ne l'ai pas fait, non.
On avait fait quelque chose comme ça à l'EXI.
Oui, je ne l'ai pas fait.
Voilà.
Ouais.
Essentiellement.
Donc, tu proposes, par exemple, qu'on doit...
Parce que sur la première couche, en général, on dit que les réseaux de rôle sortent toujours la même chose, filtre de Gabor, etc. Voilà. Alors, est-ce que c'est pertinent de mettre les quatre ou pas? Voilà, c'est pour aller un peu plus loin. Mais moi, ma question, en fait, qui va un peu plus loin que ça, outre la complexité de tes différentes contributions, mais sur celle-là, je me demande s'il n'y a pas la redondance par rapport au résidual et au gap. Parce que le résidual, il prend l'information globale d'avant, il le met dans donc il prend du local d'avant, il le met vers le global. Ouais. Parce que plus on va dans les couches, plus on va dans le global. Donc le résidual, ce qu'il fait, c'est qu'il prend un peu avant local un peu et il l'envoie à la suite. Ouais. Le gap fait à peu près ça, sauf que toi, tu prends ce local et tu l'envoies bien plus loin. Exactement. Je me demande, est-ce qu'il n'y a pas la redondance entre le résidual et le...
D'accord. En fait, là, pour le résiduel, on fait, par exemple, on crée aussi un autre, par exemple, de couche de convolution qui va prendre par exemple, ces features et va aussi prendre d'autres informations. Par contre, pour le gap, ça va prendre des informations comme elles sont. Et comme il est, et va tout le combiner avec tout le modèle gap. Donc, je pense que ça influence, par exemple, de faire de plus de convolution que de juste extraire en utilisant les gaps.
D'accord. En fait, moi, c'est surtout sur l'information locale. Ouais. Si tu prends l'information locale, que le gap prend l'information locale, il l'envoie très loin. OK. Le identity block, il prend l'information locale. Ouais. Bon, il fait la convolution, etc., max pooling et autres, et il l'envoie juste un peu plus loin, il ne l'envoie pas très loin. Ah, donc je me demande, est-ce que à partir de là, tu propages dans le résidual de l'information globale, locale qui est déjà dans le gap? Je ne sais pas. Je dis ça comme ça, mais j'ai l'impression qu'en fait, ce que tu essayes de faire, c'est prendre du local et l'envoyer vers la fin. D'accord. Mais dans les deux cas. Sauf que dans un cas, tu le propages.
Moi, comment je le vois personnellement, par exemple, si je le fais, par exemple, extraire, par exemple, un résiduel et je le concatine ici, je pense que ça va influencer un impact. Ça va influencer une redondance. Mais pour cela, par exemple, je ne pense pas qu'on a une redondance entre ça et ça.
Personnellement, c'est une question. Je ne sais pas, je n'ai pas la réponse. Hein.
Juste
parce que dans l'autre cas, en fait, tu la propages à travers les convolutions. Ça se transforme.
Oui, ça transforme.
Puisque tu as des features. Oui. Mais est-ce qu'il n'y a toujours pas cette redondance? Je ne sais pas. C'était une question que j'avais. J'avais une question sur le YOLO, mais je crois que ça a été répondu. En fait, comment tu labellises? Donc, c'est toi qui labellises? Oui. C'est pas les médecins? Oui, je suis là. Okay. J'avais une question pour le VIT. Là. En fait, tu peux utiliser VIT, mais tu peux l'utiliser d'une manière un peu plus intelligente, parce que toi, tu parlais du 3D, etc. Tu peux, en fait, utiliser ton modèle. D'accord. Et ce que tu extrais, c'est les feature maps que tu envoies dans VIT.
Ah. Okay. Donc, un modèle hybride, par exemple, s'il y a un VIT, d'accord.
Donc, ton modèle, il va prendre la 3D. Il va sortir des feature maps, c'est à toi de fixer le nom. Ouais. Et c'est ça que tu envoies dans VIT, ça sera du 2D.
Mais par contre, je pense que, par exemple, l'output par exemple, un CNN 3D.
C'est toi qui gères. Si c'est
moi.
Ouais, si c'est ton le modèle, c'est toi qui gères.
C'est moi qui gère. Ouais.
C'est ça. Créé, donc ça peut être une manière de l'appliquer. Ouais. Je pense que je ne vais pas trop tarder parce qu'il y avait une question aussi sur la taille qui a été posée, sur la taille de l'image des slides 58. pense que c'est immense. Et du coup, ton modèle, il est peut-être en entrée, je ne sais pas qu'est-ce qu'il prend. Peut-être que tu construis une grande image 2D à partir de...
D'accord. Ouais. En fait, je n'ai pas vraiment compris pourquoi elle est immense.
En fait, par rapport à ton image, là, ce qu'on comprend, c'est que tu prends chaque slice, tu les mets l'un à côté de l'autre, c'est ça?
Mais non, mais là, par exemple, on a par exemple, juste, on va par exemple oublier ça. Mais là, par exemple, on a un réseau de 2D qui est entraîné dans un batch qui reçoit un batch de 60, par exemple. Imaginez, par exemple, ou bien...
Ah, tu l'as fait comme ça.
Oui.
Donc, en fait, tu rentres chaque image.
Chaque image, oui, c'est
une image. Tu auras un batch de features.
Ouais.
OK.
Ouais, c'est ça. C'est ça pour
pourquoi je ne pas. En fait, à travers ce schéma, on ne le voit pas comme ça.
Ah, d'accord.
À travers ce schéma, on comprend que tu crées une image. Donc,
ah non, ouais, c'est ça. Ah, non, oui, là, je comprends pourquoi c'était pas vraiment clair.
Donc là, j'ai compris, tu fais un batch de la taille du nombre de slices et tu as le nombre de features.
C'est exactement ça.
D'accord.
Donc, c'est des features Mame.
Ça dépend où ils se positionnent, mais je crois que c'est des vecteurs, non?
Feature maps ou bien des vecteurs, ouais. Ce sera, par exemple, là, ouais. Exactement.
D'accord. Okay. J'avais d'autres questions, mais je vais laisser peut-être une dernière question. C'est quoi le next step? Et là, on a des classifications, on voit qu'il y a beaucoup de... Mais pour toi, vers quoi on doit se diriger? Voilà, est-ce que tu penses que la classification, il y a beaucoup de choses qui ont été faites et c'est bon, et qu'il faut se diriger vers des choses de...
Personnellement, je vois, par exemple, d'un point de vue médical, que ça, par exemple, de trouver des modèles, juste une petite minute, de trouver des modèles qui peuvent faire à la fois, par exemple, la segmentation de plusieurs composants, par exemple, du méniscus, ou bien le cartilage, ou bien la patella, ou bien le fémur et tout. Et pour par exemple, chaque région, on peut, par exemple, ça va être par la segmentation. Et pour chaque région, on va, par exemple, la classification, ou bien la prédiction, ou bien l'analyse de chaque région. Je pense que ça va avoir un impact chez les médecins. Je pense que ça.
D'accord. Ouais. Donc, mettre un peu de côté la classification et aller plus sur de l'aspect sémantique.
L'aspect
sémantique, mais cliniquement parlant.
Oui, c'est exactement ça. Ouais.
OK.
Et ça, je pense, à l'aide de, par exemple, de SAM, segmenting model, on peut, par exemple, on peut les utiliser dans cette cas. Par exemple, pour la segmentation de plusieurs composants. Et pour chacun, on va, par exemple, le focaliser pour l'analyse.
D'accord. OK. Très bien. Merci, Mohamed. Donc, je laisse mon directeur de thèse, Rachid, Jennane, Université d'Orléans, poser des questions éventuellement, répondre à des commentaires et comprendre la session.
Excellent. Qualité des échanges et pour votre contribution aussi, donc votre temps, et vous êtes déplacé aussi pour être là, pour montrer votre intérêt et puis nous donner aussi vos idées sur ce travail. Alors, comme tous les autres membres du jury, je te remercie, Mohamed, pour le travail que tu as effectué, donc pour ta présentation qui était aussi très claire, qui apporte des éclaircissements. Alors, je ne vais bien sûr pas poser de questions, mais je vais essayer un petit peu de faire juste l'historique, parce que je pense que tu as un petit peu séché sur des questions pas techniques. Là, je pense que tu as été pas bon, tu as su répondre, on verra. Les paiements du mais en tout cas, sur l'historique, en fait, quand on a démarré cette thèse, c'est-à-dire que moi, je veux dire dans le labo, mais moi, j'ai une expertise, je pense assez, j'ai encadré pas mal de tests sur la radio. Donc, je ne vais pas dire que je suis expert, mais je connais bien la radio. L'IRM, c'était la première thèse qu'on faisait sur l'IRM.
Donc, c'était le démarrage était difficile. Parce que, alors, ce que tu as oublié, c'est qu'on a essayé d'associer des cliniciens. On a fait des réunions, tu te rappelles, avec un spécial. Il nous a expliqué qu'en fait, parce qu'on avait du mal, il y avait beaucoup comment dire, de scores pour labelliser les IRM, et qui ne sont pas utilisés en clinique, qui sont utilisés dans la recherche, mais pas en clinique. Le radiologue ou l'opérateur, il regarde son IRM, il n'utilise pas les scores qui sont, que Mohamed a utilisés, qui sont utilisés aussi dans comment dire, la littérature. Il regarde, il fait son diagnostic. Il dit, voilà, il a ça, il a ça, il a ça. Mais il se refait, comment dire, il se rapporte à aucun score. Donc, qui est utilisé en recherche. On a associé aussi, tu te rappelles, Simo Saralaka de l'Université, et c'était la même chose. Donc, on était un petit peu. Donc, c'est là où Mohamed, en fait, il a quand même, on va dire au début, bûché, parce que vous ne voyez pas à travers le travail, c'est la familiarisation avec les IRM, avec la problématique, les scores, etc. Donc tout ça, en fait, ça ne figure pas dans la notation. Alors quand on l'a répété, je lui ai dit d'en parler, mais il ne voulait pas, parce que finalement, il allait vous perdre, et puis il va se faire le métro, et ça ne va pas être très fluide, la présentation. Donc il y avait cette partie-là. Donc en fait, on a essayé d'associer des cliniciens et tout ça, mais donc finalement, on s'est fait notre idée et notre expertise à travers la littérature, et on a utilisé ce qui... Alors, je ne pense pas qu'on soit à côté de la plaque, parce que c'est ce qui est utilisé dans les papiers. On a des papiers qui sont soumis, les retours qu'on a, ce n'est pas à ce niveau-là. Et puis, le papier qui était accepté, on sent bien que un des experts qu'on avait eu était comme très IRM, parce qu'il n'a pas titillé sur les modèles, mais sur les datas et sur ce qui est alors, juste peut-être pour apporter, donc, bon, Mohamed, tu as été très étonnant, parce que tu as quand même, tu t'es familiarisé avec les données, on a fait des choses, on a eu des idées ensemble. Je pense que ta maîtrise des outils, ça t'a beaucoup aidé. C'est un avantage, donc à ce niveau-là, je pense que tu étais, voilà, normal, sur le plan technique, je pense qu'il n'y a pas de souci, on va te féliciter là-dessus. Donc ça, c'est juste pour apporter un point, éclaircir un peu là-dessus. Sinon, en fait, alors je regarde, parce que je pense que j'ai balayé un petit peu ce que vous avez donné comme question sur lesquelles un petit peu rester comme ça. Je pense que j'ai un peu... Oui, je pense que dans ce que j'ai dit, j'ai pas mal regroupé. Ah oui, donc en fait, pour les paramètres, il y avait cette question-là sur les paramètres statistiques qu'on a utilisés. Alors, c'est vrai qu'on peut toujours rajouter des paramètres, bon, mais généralement, on utilise la sensibilité, on utilise la spécificité, et tu les as en plus. Parce que pour calculer l'accuracy, tu les as. Donc, il faut décider de les ajouter. Alors, on ne revient pas à l'idée. Pourquoi on ne les a pas mis? Parce que ça permet quand même, ça donne des indicateurs sur ce qu'on peut, qu'est-ce qu'on rate, est-ce qu'on rate les faux positifs ou les faux négatifs. Est-ce qu'on aurait plus raté? Oui, donc ça, je ne sais pas. Donc c'est quand même un peu important qu'on les a. Alors, on ne les a pas mis, je pense que c'est par. Ça ne nous a pas été rapproché par les experts sur le papier. Enfin, en tout cas, c'est le papier qui a été accepté, les autres, on verra. Mais en général, on aurait dû les mettre. Alors, il y avait la dernière question qui est intéressante, c'est celle que tu as posée, Sylvie, c'est sur est-ce qu'on a fini, même si on atteint 99%. Alors, en fait, là, c'est très simple, en fait. Il faut savoir que nous, ce qu'on essaie de faire, c'est apporter un outil d'aide au diagnostic. Ici, on va voir les radios, on leur dit on a un outil qui fait 100% du travail, il faut être sûr qu'ils ont accès, ils ne vous en voudront pas. Ils ne voudront pas, en fait, eux, ils veulent avoir le dernier mot, ils veulent un outil d'accord qui les assiste, mais qui leur dit pas, voilà, ça répond parce que, bon, déjà, ça veut dire qu'ils ont plus rien à faire. Mais bon, on est loin de ça. Et il faut savoir que je pense, pour ce qui est du cancer de la peau, je crois que les Google, je ne sais plus, ils en fait, il fait plus de 99%, mais il n'est pas cherché, ils ont fait beaucoup de buzz autour, mais il n'est pas pour autant utilisé, il ne va pas remplacer les autres. Et de manière générale, il y a aussi, ça je pense que c'est un constat qui commence à être fait au niveau des conférences médicales qui sont sérieuses et tout ça, c'est-à-dire qu'il y a beaucoup de buzz autour de l'IA. Il y a très peu de modèles qui fonctionnent déjà des choses, mais il n'y a pas vraiment, donc il y a beaucoup de choses, on parle beaucoup, il y a fait, mais il n'y a rien qui fonctionne vraiment. Bon, il y a des choses, c'est sûr, des choses qu'on ne voit pas, mais il n'y a pas vraiment il y a beaucoup de buzz, mais il n'y a pas de réalité clinique qui utilise vraiment l'IA, etc. Donc ça va venir, je pense, mais pour l'instant, je pense que c'est plus. Donc, merci à vous tous, merci à moi-même. Bonjour, la parole à M. le Président.
Merci Rachid pour avoir complété un petit peu les questions. Donc là, on va passer à la séance où le jury va se rassembler et discuter un petit peu de ta prestation et de tes travaux. Et on va par la suite délivrer. Je ne sais pas, Rachid.
Et donc, il y a un point, moi, la prestation.
Oui, j'ai avant de terminer.
Oui, mais je vais juste
terminer. Les docteurs ont le droit de poser une question, si vous voulez. Donc, une ou plusieurs. Hein. Pour ceux qui souhaitent, qui ont un doctorat.